Thank you for joining us for our second Integrated Pest Management webinar. And this pest management webinar is focusing on pest monitoring and identification. I'm Colette DePhelps, and I'm an area extension educator and one of our Idaho Cultivating Success team members. Cultivating Success is an educational program that has been doing in-depth education and webinars for small and mid-sized farm and ranchers for over 25 years. The program was started by University of Idaho Extension, Washington State University Extension, and the nonprofit Rural Roots. So we're really glad to have you participating today. And we want you to know that the recording of this webinar will be available on the Cultivating Success website later this week. We will also post the slide deck for this webinar. Just a couple of tips. We are not in meeting format. So meeting format is when you get to see all of the participants. We're in webinar format, and that gives us a little bit cleaner of a start to our webinar and end for that online recording. So if you are having any trouble, you can type in the chat at any time. Kate Wood, our program coordinator, is standing by to help you with any technical assistance or any technical questions. Also, if you have any problems with your sound, you may want to go ahead and switch to the phone. You can do that through your audio button. You just click on that, and it will give you a phone number to call in, and then you can mute your computer so you don't get any feedback. At any time, you can type questions for our speaker into the Q&A box, and we'll have time throughout the presentation to be able to answer those questions. I know that Desiree will probably also ask you to post in the chat. So if you want to find the chat in the Q&A box in your control bar, this is a good time to do that. Again, the webinar recording is going to be available on the Cultivating Success website with the slide handout. With that, I want to thank Desiree Wickwar for joining us today and for being our lead presenter for this integrated pest management series. So I'll go ahead and hand this over to you, Desiree, and we will start with a poll just to get a better idea of the level of confidence that you all are coming to this webinar with. So Kate, if you could launch that poll. So you will see a couple of questions on your screen. So how confident are you in identifying pests? And then how confident are you in monitoring pest populations? So we see that we have almost all of our participants who have completed the poll. We'll just give it another second to wrap up. Okay, Kate, let's go ahead and end the poll and share the results. So what we can see out of our 18 participants that completed the poll, one person is very confident in identifying pests, and the majority of folks, about 80% to 90%, are somewhat confident, and one person is like, I am not confident. That's great to know. Thank you for completing that poll. We hope that we are able to increase some of your confidence by the end of today's webinar. And then looking at monitoring pests, we see that a couple people are very confident in how to monitor pests, but most people are not confident or somewhat confident. So that's a really great baseline for Desiree to know as she moves into the content of today's webinar. Again, thank you for participating in that short poll. So Desiree, thank you for being here today, and the screen is yours.
All right, thank you so much. Those of you who were here earlier this week might have already heard this, but I managed to break my ankle this week, so I'm working from home with a little bit limited bandwidth, so that's why my my video is off, but I'll share my screen now. Hopefully we won't have any bandwidth issues. So, okay, yeah, thank you all so much for completing that poll. That is really genuinely so useful, and we will talk maybe more extensively about monitoring than about identification today. And it looks like that might befit everyone's needs. So yeah, I think Yeah, so a couple reminders on getting pesticide credits throughout this series. Just note that you have to be properly registered. You have to have input each individual's pesticide applicator license that you want to get credits. So if you have two people watching the same screen, you need to have separate registrations, and then we need to be able to document with ISDA that you were present for the entire duration of the webinar. Um, that's just their requirement. So I need to have every participant registered and every participant who wants credits in our Zoom roster on a separate device. If you have any questions about that, I can address them at the end of the webinar. And okay, getting started. So as Colette mentioned, today we're talking about both pest monitoring and pest identification. So we'll start with a little bit more pest monitoring. Again, for those of you who are here earlier in the week, we kind of briefly skimmed over some of these ideas, but today I'm really going to delve in a little bit deeper, particularly on a couple key pest monitoring tactics. So, just in case anyone wasn't here for our first part of this series, where we talked about what the definition of IPM is, or just in case we need a reminder, I'm gonna go through that really quickly. So, when I think about IPM, I think about it as a quote unquote systems-based approach. So, what IPM aims to do is not just look at a pest alone, but to look at a pest as an entire part of an ecosystem and understanding how these ecosystems, even if it's a highly managed ecosystem, say a greenhouse or, agricultural field or whatnot, even those highly managed ecosystems, the pests are a part of a bigger system, and IPM aims to understand how all these pieces and parts of an ecosystem work together to influence that pest and influence how we need to manage that pest. So when I think about IPM, I think about it as this sort of process where we're first and foremost monitoring what's going on in our system, fully understanding, like I said, all those cogs that fit into our ecosystem machine. Then based on that understanding of an ecosystem, we decide, is an action threshold met? And we'll have a little bit more time than we did earlier this week to talk about exactly what an action threshold is and how you might determine that. Um, if your action threshold is met and you need to move forward with management action, IPM then lets you select amongst a wide variety of treatment tactics. Again, kind of understanding the ecosystem to help you better pick the best treatment tactic for your pest problem and treat things the most effectively as possible while minimizing risks to the ecosystem and human health, and minimizing costs as well. Okay, so here we are on to pest monitoring. So everyone kind of has an idea of what monitoring pests, right? Is we're just keeping track of the pests, but what exact kind of like data? What is the nitty-gritty look of that, or what does the nitty-gritty of that really look like? Of course, we have observing pest presence and absence. That just says, is this thing here or not? That can just be throughout an entire season. Do you ever have a pest pop up that you need to keep track of for future seasons? Or that could be if a pest pops up at a certain time. That sort of gets into observing the life cycle of that pest. Like I mentioned, IPM really asks us to do a deeper dive into the ecosystem, biology, ecology of the systems that we're looking to manage. And understanding the life cycle of a pest can really help you understand when it's going to pop up. And as we all know, that makes a huge difference in how and why we determine or how and why we manage. Observing population dynamics. That's saying, how is this pest population growing or shrinking? How rapidly is it rising? Is there something going on in the ecosystem that's going to allow it to absolutely explode? Or is there some maybe abiotic weather condition that's going to dampen down the growth of that pest? That's what we're keeping track of. This interplay of abiotic and biotic factors that again is really just fundamental in understanding an ecosystem. And I know I harped on this a little bit last time, but I would be remiss in not reminding us all that understanding natural enemy populations and their effects is another quintessential part of monitoring. I even made the mistake of putting pest monitoring up at the top of my title slide here, but what we're really doing is just monitoring the ecosystem and understanding how these natural enemy populations are doing some work for us is another part of this. So when you're looking at all the tactics that we'll talk about today, and even some of the models, keep in mind that there are some tactics and tools and whatnot that you can use to understand your natural enemy populations and kind of how they're exactly going to interface with a pest population. So our overall goals all this data is supposed to tell us the level of incidence. That can be either abundance or density. There's different times when we're more curious about one of those factors than another. Remembering abundance is just the sheer number of a pest or really any organism that's around, and the density is the number of a certain pest per unit area or volume. So that's what we're looking for. The loss caused by a pest. This is another thing we can kind of keep tabs on as we're monitoring. Let's say we know the level of incidence. We know that there are, because I'm an entomologist, I'll pick some of my favorite examples. Let's say there's a certain number of Colorado potato beetle larvae per pepper plant, and they are causing 50% defoliation. That can help us translate pest presence to what we're actually interested in, right? Which is how it affects us. We're not interested in the pest just for the pest's sake alone, but for that loss. And then, of course, the economic benefit that control would provide. So that's saying, taking these first to two steps, the level of incidence, the loss that could be caused, and saying, okay, is it worth making a treatment? And so I think you can see here exactly why monitoring is so important in that process of IPM because it gives us the data that we actually need to undertake that process and manage pests effectively. Okay, so we're gathering all this data, but once again, we're not interested in just the pest itself. And we're not interested in, okay, exactly what is the level of damage right now, because what's done is done. What we're actually also interested in is how that translates into the future and how that suggests what we should do right now to prevent a certain type of change that we don't want, probably increased damage. So how we do that, of course, is we take our surveillance data, our monitoring data, and we turn it into forecasting information. So what we're looking to do there is predict pest outbreaks. Again, that can come from when exactly is a pest going to arise? Maybe, for certain pest, it might be the case that inevitably, if it is present at a certain time in a certain setting, it will cause damage. Um, maybe in a seed potato field, what you might get is a aphid outbreak. Well, that's unacceptable because they transmit viruses. Um, or there might be another way you might be interested in predicting a pest outbreak is just predicting if it's going to hit really damaging levels. So both of those things are elements we're looking to predict. Presence, absence, but predicting the need for control measures. We'll get into some of the tools that we have at our disposal to get at that a little bit later, and predicting suitable timing of control to achieve maximum production. This is a quintessential one, right? Like, I think maybe everyone here has, to one extent or another, applied a pesticide and maybe not achieved the exact level of control we were hoping for. So frequently, that can be a result of the fact that the timing was just a little bit off. Many pests have a really specific stage at which we are looking to target them to prevent them from escalating to uncontrolled numbers, or they might have a specific stage in which they are only then susceptible to the pesticides we have at our disposal to treat them. A great example might be, again, I'm an entomology biased person, but maybe we have certain weevil larvae that develop within the stem of their host plant. So what they're doing is they are eating out the inside of that stem. That's causing damage, but unfortunately, they are extremely well protected within that stem. And so even if we were to go out and apply a double rate of some insecticide, if we can't, and it's a contact insecticide, if we can't get to that larva, it doesn't matter. So predicting exactly when we need to apply our control measures, whatever those are, even some of our non-chemical control tactics. Predicting suitable timing is also a key part of monitoring, reminding us that we need to know exactly what's going on in pest life cycles in biology to adequately manage them. Okay, so types of pest forecasting. Again, this is just kind of getting at these general ideas. There's short term, of course, that's based on maybe 1 or 2 seasons or just within that one season. That's saying, what is going on right now, what's going to be going on in a week. And long term forecasting, that's based on the effect of weather on a pest. So maybe what we would do is take a 30-year average of weather events and climatic variables to understand how a pest population is most likely going to behave in a year. And we'll look at some data um, that shows both that short and long-term forecasting here in a little while. So the tool that I've been sort of obliquely referencing periodically here, where I say, okay, we're going to have monitoring tools, the one that we're really going to dig into today is the use of degree days and degree day modeling to understand progress, pest progress through their life cycle or their phenology. So what is a degree day? This is sort of a confusing term. Um, a degree day is a measurement of accumulated heat over time. So it's not just a period of time, and it's not just an amount of heat, but it's sort of this conglomerate unit that's helping us understand how heat is accumulating over time in a given season. Why do we care about that? Well, many pests rely on these accumulated degree units or accumulated heat for their development. Insects, again, I'm biased towards them, but they are a great example of this. We know that insects are quote unquote cold-blooded, right? And what that practically means is that they need heat units from the outside environment in order to go through their life cycle. So, what happens, maybe we've seen this with something like spider mites. What happens is if it's really warm, they accumulate those heat units really, really quickly and they go through their development much more quickly. But if they accumulate heat units really slowly, we see much bigger lag time in their population growth, right? So what this figure is showing here with a potato psyllid specifically is that what we know is that a specific number of degree days, aka heat units, are necessary to get a psyllid from that egg stage to this larger nymph stage you see in the middle, and then a specific number are needed to get that nymph to grow out into an adult. And there has been a truly prodigious amount of research that's gone into understanding exactly how many degree days are necessary for different pests to pass through different developmental cycles. It's really different based on the region and different species and different subspecies. Um, there are many caveats with using degree days. If we develop a degree day model that says you should expect to see a codling moth pop up at this day, but that is, from data on the East Coast, well, that might not be relevant for us out here. So though I think degree days are an extremely powerful tool, I think they do come with a number of caveats. And so that's why we're going to do a bit of a deeper dive into understanding how to use them today. Because if you try and blindly use them and sort of take them as gospel, you might be doing yourself a disservice. That said, it is really, really powerful the amount of research that's been done into just getting these numbers out there that says, here's how much time under what temperature conditions it's going to take a psyllid to go from an egg to a nymph. That's really actionable information for us. So what do we need to calculate a degree day? Essentially, we need the maximum and minimum temperature in a given day and a upper and lower development threshold. I guess I should go back. Maximum and minimum temperature in a day, That's to allow us to just understand how many thermal units we're developing in a given day, right? And then we can stack those up over days or weeks or months or years for some species. We need upper and lower development thresholds. What that means is, again, we'll go back to that psyllid. Um, you might have really low temperatures, as you might expect. Maybe no development can happen. There's just not enough heat for that psyllid to continue growing and developing inside that egg. But there's actually sometimes a upper development threshold as well. And that means, like, let's say it gets extremely hot, maybe that development of whatever organism we're talking about also ceases. There's just too much heat stress, and things really completely slow or stop. One thing as we get into looking at these models specifically, sometimes there is no upper threshold given. A lot of insect pests in particular are pretty temperature tolerant. This is less true for diseases. Um, but for certain models, there's no upper threshold developed. This is in part because usually, none of these degree day models are going to tell you something down to the minute. They're the best approximation we can get, and oftentimes they're accurate within a day or two when they're predicting different developmental stages. But as with everything, data and models are just modeling reality, and they're not entirely accurate. So sometimes an upper development threshold isn't given, because it's really time and research intensive to develop one, and usually you can kind of get away without it. Is the short answer, but great models do have them. And then a biofix date. What does that term mean? Essentially, a biofix date is the date at which, in a calendar year, we would expect development to actually commence. So this can look like a few different things. It could look like a biofix date would be, when the egg was laid. It cannot accumulate degree day units before that, right? It can't develop because it didn't exist yet, just based on the developmental timing in the life cycle of an insect, or pest. Another thing it could look like is sometimes we arbitrarily set a January 1 biofix date, kind of like arbitrarily setting an upper development threshold or something. But that is data that we need in those models. Oftentimes the model will provide it for you. But if you're trying to understand it on your own, you might want to have a general idea of, okay, when would this pest reasonably start to develop? Sometimes it's just early spring. Okay, there are different ways to calculate degree days, and some of this is a little bit academic because chances are you don't really need to ever do this for yourself, but maybe this will help us understand degree day units. So one method we have is the average method. And as the name implies, and you can look at this little equation I have on the screen if you're really interested, the name implies, basically what we're doing is we're taking the daily maximum temperature, adding the daily minimum temperature, and dividing by 2, that is to get an average, and then subtracting our lower development threshold to get a number of degree days in a given day. And then these would be added up over time. A great perk of this is, particularly before we had really impressive computational power, it's pretty easy to calculate. If you're just trying to get a rule of thumb to say what's going on in your system, you can do this. Unfortunately, it is definitely the least precise of the methods, and certainly less precise than the other method that we'll talk about today. As you might imagine, there's a lot of fluctuation in a temperature over time, even in a day, that the average really just doesn't capture. So you can have a lot of noise. A great way that we can calculate degree days is using what's called the sine wave method. And again, we're only going over this so we can understand the underpinnings of the models we use, because you won't be having to do this on your own. Fortunately, it's been done for us in many different venues. But the sine wave method essentially is trying to get away from what the average method lacks, which is kind of just taking the average to get a general idea. So um, basically, what you see here is a sine wave. So it's a wave that's created between, let me see if I can figure out how to turn on my little pointer here. It is Here we go. Okay, so what you see is this um, minimum. Oh, it didn't work. Okay, well, I can't seem to figure out the pointer. But anyway, the minimum temperature is given in these black dots and the maximum temperature is this. So we would kind of assume that there's going to be a general upward swing towards the max temperature in a day and a general downward trend that kind of looks like this. And that's just a model of a temperature based on data points of the max and min. What this might actually look like is, of course, a little bit different. Like, let's say, this dotted line here might represent the actual change in temperature over time for 2 days. This is what this would be. So we start at night over here on the far left, and then we would have maybe a little bit more rapid increase in temperature than the model would suggest, and a little bit more rapid decrease over time. But in general, what you kind of see is this tracks pretty closely to how we would actually model the temperature. Why we don't take an infinite number of points is one, because usually we don't have the data. Um, and yeah, usually we don't have the data, and it would take a lot of computational power to count up or to understand degree days should we use a more exacting method. So when we're using the sine wave method, what we're trying to do is we are modeling the temperature change, and then the area under the curve, maybe you remember this from math class way back, the area under the curve is what we would call the accumulated degree days. So in this figure, you see again our modeled temperature curve with minimums and maximums given in a 24-hour period. And I should say these are usually data points that we get from a weather station, for instance, or you can take them manually if you're doing the average method. Then what we insert into this graph is a lower development threshold. So this would be a representation of, like I said earlier, sometimes we don't have an upper development threshold, but we cut off this bottom portion because we say, okay, below this blue line here on our where it meets our y-axis, there can be no development occurring. It's just too cold. The nightly minimums are too cold for development to be happening during part of the day. But then, during the vast majority of the day, what we see is that the temperature is above that lower development threshold. And so degree days are accumulating, and that species that we're interested in is developing. Okay, let's say we're using a model that has an upper development threshold. How does that change things? All it's basically doing is saying during the very hottest middle part of this day, there is no development that is able to occur. It's too hot and too stressful. So that takes off the top of both of these curves for this model and says, okay, we're no longer counting the area under that part of the curve. So then what you end up is these two gray shaded middle sections would be how we're accumulating degree days. So yeah, area under the curve. That means we sum all this up, over all of the days since a biofix, and that says what our accumulated degree days is. And I'll show some tables that might help clarify this point a little bit. Sine wave method, like I said, is definitely the most precise, most complex, but it is the most complex to calculate. Like if I was to go look at this and be like, okay, great, just go out and take your minimum and your maximum temperature and just calculate the sine wave, add up the area over the curve for, like January through May. Just do every day, no big deal. No, it is a big deal. It's computationally intensive. So we're going to now, I'm gonna bop over to a website that I will also paste into the chat here that is a really great tool for looking at different degree day models. There's a ton of them out there. I'm a little biased towards this one because but Utah State also has a great interface for this too through their climate center, and different universities and whatnot have some other great tools for this as well. So these degree day calculators allow us to get that sine wave method of calculation. Usually that's what the model's using, to know when pests are going to be going through certain stages, like we talked about earlier, that can let you know when to apply, what to apply, if you're going to hit certain thresholds as well. And then importantly, some of these models also have integrated into them suggestions on when to monitor. So, a degree day model might say something like, okay, during when this many degree days have accumulated in a year, we would expect 30% of eggs to have hatched of this pest, and that's when we recommend a systemic spray to, target those larvae as they're developing, whatever that looks like. So if you're seeing recommendations in publications that you're looking at for pest management that say, treat when 30% of egg hatch has occurred, then you're like, well, how do I figure that out? Degree days are the answer. All right, and with that, I'm going to see if I can share a different screen and we'll go walk through the site itself. All right, so this, I will admit, part of my reasoning behind wanting to walk through this with you guys is because I certainly recognize that it is maybe not the most user-friendly interface. The guys who develop it are big nerds with big brains, and it can be a little bit challenging to understand. But with a little bit of practice, I think this can be really useful. So when you go to the website that, let me think, yeah, I put it in the chat. So when you go to this website, this is what you're going to find. I put the original homepage because if you really want to geek out about this, there are some ways for you to sign up for email notifications about different phenological events or phenology, what you see here. That just means the timing of pest development events. So you can get these email notifications. Let's say you manage a specific pest. You can see this push notification here. I think you guys should, this guy. And you could sign up for that if you manage a specific pest and you're going to be worried about it. You can also get these risk models that can help like maybe a more complex understanding of an ecosystem that you're trying to manage. But what we're going to delve into today and what I personally use is this second link here. It says Degree Day Model App. It's a mobile-enabled interface, and personally, I just think it's a little bit easier than the web interface. I'll click on the web interface just for a second. You can see there's a ton of different options here, and you can largely do a lot of the same things. If you really want to get into nitty-gritty, you can delve into that, and I'm happy to talk with folks about it. But what I would recommend using is this mobile-enabled entity that's been developed more recently. So what you're going to see when you click on that site is, if you want, you can read an introduction. If you kind of forget some of this, you can look at this instructions for use tab or the station tab that can help you understand exactly where data is coming from and remind you of exactly how to best use this interface. But hopefully I can do a little bit of a proxy here today too. So we'll go through just like an example here. What would I do? So first off, what you're going to do is, let's say I have an apple orchard. And what I'm going to do is I'm going to try and find a weather station in this network. There's hundreds and hundreds, I don't even know, maybe thousands of weather stations linked to this site that you can be pulling data from. If you're a real nerd, you can upload your own file, but what you're going to basically doing is trying to find the weather station that is closest to the place you're looking to manage. So let's say I personally am in Jerome right now. So I'll search for stations there. Let's say that's really close to the orchard that I'm looking to monitor. Say I'm in Jerome County. Okay, here's my options. There's a few different weather stations. I see just Jerome County Airport, the Hanson Bridge, the Perrine Bridge. How exactly do I choose one of those? Well, what you're going to want to do is, of course, first and foremost, I would say pick a station that's really close to you. We know that there can be a ton of variation in our data just based on pretty small geographic differences, right? So what I would try and do is pick the station that's closest to me. What you're also going to see is this quality score. What the quality score is getting at is it's sort of this like compound way of taking a bunch of different data about what the weather station is providing for a given model and saying just how good it is at serving that model. Sometimes it'll take into account things like missing data or if it has, a really frequent report of each unit or really infrequent. The general rule of thumb is that most of the stations that you're going to see pulled up are filtered to be the high quality ones. Um, but in general, you're probably going to want to pick the one that's closest to you as long as it's not, immensely lower quality score than the others. My rule of thumb is like if they're within 10% of each other, then you don't really need to worry about their difference that much. So I'm pretty close to the Perrine Bridge. I'll select that one. Okay, cool. I can see currently selected Perrine Bridge. So that's the model or the station I want. Okay, now I'm looking to not just understand a particular location, but a particular pest. So then I click over to this model tab that you see in the center of those tabs here. So again, I said we're pretending I have an apple orchard. I wish I did. So I would say, okay, what I'm looking for is going to be models relating to apple and pear. Drop-down menu. These are going to be all of the different models that are available to select from that have been loaded into this interface. Once again, if you want to really delve into this, a lot of these will give you um, a, a publication that these are derived from. For instance, you see apple maggot first emergence. That comes from a paper that is Jones et al. 89. If I was to click on this, I could see that paper. And if you want to analyze it and say, wait a minute, exactly where was this data gathered? So is it going to be relevant for my location? You can delve into that and make sure that all the data is from your specific region. But let's say, oh, and you probably want to try and pick a newer model because we know that pests change, varieties change, our economics change. So in general, a newer model is probably going to be better. As with so many things, there's a lot of caveats to that, but just so you know. All right, let's say I like the look of this. It's a very new model. It's from WSU, and it doesn't ask me to have a biofix, apparently. So I say, okay, WSU, that's pretty close to us, maybe especially for those of you who are up in the Panhandle. Um, and I'm interested in codling moth. So that's the model I'm going to suggest or select. So I guess one thing I should say, this particular model doesn't ask me to select a biofixed date, but if you did and you don't have one, when in doubt, just leave it as January 1st unless you have specific guidance. Otherwise, that's oftentimes fine. Okay, cool. So um, there are some other options that you're going to see down here. NMME extended forecast, that's just a particular blend of weather models that's going to, you know, um, be like an extended forecast for the weather at that station. This is a different seasonal forecast. This one's the default. You can look at a 30-year average. I'll select a 30-year average because I'm going to say, okay, I just want to know kind of what goes on average. If I scroll down here after I select that, I can see a plethora of different information. If you really want to delve into the nerdiness, you can say, okay, cool, this is good because this is used that sine wave method that we talked about. It's not using average method, so it's a little bit more exact. Okay, great. Also, this model has a specific lower and upper threshold that it has developed. Maybe I should zoom in on this. Yeah, there's a specific lower and upper threshold. So someone went out and observed that. Model is really cool because it uses a whole separate model to determine a biofix date. So that's when that development can start happening. Um, but as usual, it defaults to January 1. Okay, so that's what's most important. You can note that the region of known use is in the Pacific Northwest. So it's saying, okay, cool, people in the Pacific Northwest are using us. That's us. So that's what we'd expect. Okay, now what is this model providing us with? It's saying again the number of degree days. So we see that over here, for instance, is 250 for an expected life stage event for a pest. So for instance, at 250 degree days accumulated in a year after your biofix date, so after the start of development, we would expect to still be in the first generation of coddling moths, with just 3% of the eggs having hatched. And then you can see as we go down here through subsequent generations and developmental events, this is the data that this model is going to be pulling from. So once again, if you're seeing a recommendation that says, apply X at a certain number of degree days, or even more likely, apply this compound at 30% of egg hatch, this is kind of a place you could come to find some really great detailed information about that. Um, and you can also, even if you don't have a publication that gives a specific number like this, you can say, okay, well, I know I don't want to apply this when 75% of the eggs have already hatched because maybe they have burrowed into the soil and whatever this pest is, maybe it burrows into the soil so they're no longer contactable anymore, and you want to apply before the eggs hatch so you can target the eggs. That's just an example, not relevant for codling moth, but yeah, so you can use this to kind of understand this is the 30-year average, but you can kind of understand what life cycle events you would expect to happen at certain times. Okay, output. This is going to say again for that 30-year average, the date. So this is like, just a calendar date, like April 30th. The expected maximum and minimum temperature, um, and then the start of development, for instance, is right here on 4 of 30. So that's more specific data. You'll notice that if I go back to my model and I check the NMME extended forecast, that's going to try and forecast for this year. One thing that's kind of interesting is you can show a date comparison table. Something we oftentimes hear people say is like, wow, we're like a month ahead compared to last year or something. There is actually some data on this. So versus 2024, this model thinks that we are 4 days ahead of what we saw in 2024. Compared to 2023, we're 28 days ahead. And compared to our 30-year normal this year, we're 10 days ahead of development. So that's actual calendar days, not degree days, but that's based on degree days, right? It's saying, wow, in 2023, we had accumulated way, way, way less degree days by this time of year. So development is going to be significantly expedited compared to what you saw in 2023. So that's kind of helpful as well. I will admit that I think that the table is perhaps the most useful output, but you can see this in graphical format as well. So you can compare different observed data points and different observed data from this year and expected forecast data. For instance, that anemone blend of forecasts is, again, this is not the most useful. But yeah, you can find that here and you can start to find the split. So, you could say the 2023 observed line is this one, 2024 observed is this one, and you can start to see certain differences in those models as they go out through our growing season. I think that is what I have to say about these. I will switch back to my other screen now. Thanks for bearing with the screen switching.
I just want to jump in and let everyone know that if you have any questions, you can definitely type those in the Q&A box. If you have any trouble finding the Q&A box, you could put them in the chat.
Oh, yeah, that's great. And actually, I think if memory serves, we can take a little pause right here to take any questions on monitoring or let's see. I'm not seeing the Q&A box. Maybe that's just because I'm in screen share. Colette, do you see any questions so far?
I don't see any so
Okay.
far. I just wanted to remind people that they can type them in at any time and then we'll pause. So
Okay.
while we don't have any at the moment, uh, just definitely if you have questions, please type them so that we can take a pause and answer them.
Yeah, that would be perfect.
Okay.
Okay,
Oh,
yeah, I know that was a lot of information. Hopefully, you can, if you're interested, you can dig around on that site and it just takes running through things a few times to kind of understand that and hopefully you can glean something from it. So, okay, I have a little bit more information on pest identification and then we'll wrap up. Okay, so um, I went through this a little bit earlier this week. So, just as a reminder, let's say you're going out, you're doing your monitoring in the field. What's the process you should be expecting to identify a pest problem? Let's say you're not going out and looking for a specific pest, but you're just saying, okay, what's happening in the system? Remember first to identify, of course, what plant. If you're in a monoculture, that's not relevant, but let's say you're in a garden identify the plant, identify the symptoms, but then really remember to look for potential environmental stressors that could either be attracting a pest, maybe, drought stressed plants, invite certain pests to attack them that can really help you narrow down what pest might be most likely as you're working to identify that list and sort of identify a culprit. Looking for the pests themselves is of course a great example of helping you identify a pest, but look for their sign as well. Like if you see frass, which is insect poop, you can say, okay, I think this is probably coming from this certain insect. It's not a disease, or different symptoms, for instance, from a virus. So keeping tabs on those. And when you're going out and you're identifying your pest, just reminding yourself to determine their distribution and severity. This is sort of double duty monitoring and identification. But sometimes the distribution of pest damage can be a great tool to help you figure out what it is. Again, you've probably noticed I like talking about spider mites. A great example for spider mites, spider mite damage often occurs really heavily right by dusty roads. So you might be saying, what the heck is going on? I can't see any pests because they're really small. Spider mites are. But what you might be able to say is, wait a minute, like it's only the plants right by a dusty road that are seeing really heavy damage. That might clue you in too, knowing that it could be a spider mite problem specifically. So all this data can really help you narrow down for yourself or narrow down with an expert what pest you might be dealing with. Do's and don'ts. In the interest of time, I'll skip this, and since we went over it, just try and use um, reliable resources. Don't try and rely too heavily on identification apps. You can use them to get to something coarse, but just know that sometimes they're wrong. I wanted to share a few identification resources. Maybe I'll go ahead and copy and paste this into the, whoops, that didn't work. I was gonna say I'll copy and paste this into the chat, just so you guys have it.
okay. We do have a question that came into the chat.
Oh,
Pat's
great.
asking, can you use the upset site for beneficials and for pests?
You can. That is a great question. And because I think it's well, yeah, let me see how hard it would be for me to share this. Let me share my that same screen. So when you're selecting a model, um, you can select um different beneficial insects. Yeah, they're right here. Different endangered species. Thanks for reminding me to go over this. Different invasive insects, plant diseases, all kinds of things. Beneficial insects is, of course, a great example. There's not to date, but we're getting more all the time. And some other, I'm trying to remember if the Utah State, um, site has more of these beneficials, but you certainly can use it um, to look at what's going on for your beneficials as well. There's degree day models for that in a bunch of different places. I think that answered that question. Beneficials WordPress. Yep. Yeah, and it is for disease too. I apologize for my insect bias. I know. It certainly is there. Okay, so I put these in the chat. There's some great resources. I'll kind of blip through some of this here at the end. Insect Pest, this insect ID site at the top allows you to submit photos to experts here at UI and will help you identify diseases. Our diagnostic lab is great with that. You can submit samples and photos and just get in touch with them. You can browse different pests on our IPM website. And then I included some other just that I happen to be familiar with for cereal pests and invasive insects. Um, but there's just a handful of identification resources. As usual, I don't have the time to go over all of them, but there's so many out there. Just try and use something reputable and maybe not from a company if you can. Okay, I'm running into the same problem I did last time, but I wanted to, since I didn't do action thresholds as much service as I wanted to in our last webinar, and maybe some folks are new anyways, wanted to go over briefly what an action threshold is. So when we're monitoring, like we said, we're not interested in the monitoring itself. We're interested in determining if we have to do something about what we find in our monitoring findings. So that's where action thresholds come in. And as with degree day models, there are a number of caveats to their use. They're an amazing tool that can help us make smart environmental and economic decisions, but only when we understand that, just like with a degree day model, the data for an action threshold comes from a specific study that was done in a specific place in a specific time. And oftentimes researchers will do their best to make these as universally applicable as possible and to make them age well. But yeah, just understanding where your action threshold data is coming from and maybe kind of using your own knowledge and consulting with experts to understand how you should use that data can be great. So what am I talking about? What is an action threshold? Action threshold is when you should take action to manage a pest. Um, we kind of talked about this last time, but this can be based on the economics, saying, oh, this pest is causing so much damage that it's going to cost me less to treat for it. Or it can be just saying, gosh, this pest is causing so much visual damage that that is no longer acceptable, and we really need to do something to stop it from ruining whatever setting you're in. Bunch of different ways to do that. But this is what I had to skip over last time that I wanted to touch on. So in helping us understand the crux of using a threshold, this is the data that we're looking at. So what we see on our y-axis here is the pest abundance and x-axis over time. So how is the pest abundance changing over time? What we see in this top line is an untreated population, a scary curve to all of us, right? Like what we don't want to see is a pest population just exponentially exploding until it hits its carrying capacity and is just eating all your plants. Um, what we want to see is treating that pest before it gets to that point. But how do we select the point on that graph? Well, the point of treatment that we pick is right about at the action threshold or the economic threshold. And that's saying the pest has started to inflict more damage than we can accept. And that's before we hit our economic injury level, which is just to say that the pest has caused an unacceptable level of economic damage. So that's kind of what that point of treatment looks like. And since I didn't provide the definitions of the terms, actually, I'll nix this slide too. So hopefully we can leave some time for questions since I ran a little over. So anyways, there's a rapid run through some information on monitoring and pest identification. Hopefully, I lingered on the monitoring aspect of things since it sounded like we had some uncertainty with that. And monitoring really is maybe the more black box of identification. Usually with identification, you can send in a photo or send in a sample and get a good definitive answer. And we have really powerful web-based tools. But monitoring can be a little harder to be able to understand what's going on. So hopefully, understanding degree days and some of the other principles behind monitoring can be helpful. And even if you don't have a specific model that you'll now use as your treatment gospel for a pest, you can kind of go and help yourself understand what different pests are doing at different times of year with those degree day tools, or understand them if they pop up in different publications. So, yeah, any questions?
I haven't seen any additional questions come in, Desiree. Thanks for that thorough presentation. Again, this has been recorded. So if you want to go back and, review what Desiree has shared today, it will be available on the Cultivating Success website. We do have another question from Tad that just came in. Is there a place we can pull info for beneficials to pest ratios, like cereal leaf beetle to ladybug?
That is an excellent question. I'm trying to think of what I know about. It can be challenging because there's so many different combinations, right? Um, I am not aware. This doesn't mean it doesn't exist. I'm not personally aware of a repository of information that says, this many lady beetles will remove this many cereal leaf beetles from your monitoring equation that we've been sort of talking about. Sometimes you can find it in specific publications, but um, yeah, there unless you find it, in a specific fact sheet or whatnot. I'm not aware of any repository like the US Pest for degree days.
Thanks, Desiree. Any other questions? Go ahead and type them into the chat or the Q&A box. Well, while we wait to see if there's any other questions, we do have a couple of questions for you. Kate, could you launch our final poll for today's webinar? So after participating in this webinar, how confident are you now in identifying pest populations, and how confident are you in monitoring pest populations? So are you a lot more confident, somewhat more confident, or really no change? Okay, we just have a couple more people to participate. Give it a second. Okay, let's go ahead and share our responses. So in terms of identifying pest populations, we have a few more people that feel a lot more confident. The majority of our participants today feel somewhat more confident, and a couple of people feel that there's no change in their confidence. So great. Thank you for sharing that. It's great to know that the majority of people feel more confident leaving the webinar. And then looking at how confident you are in monitoring pest populations, we have almost 45% of people that feel a lot more confident in their ability to monitor pest populations, and another 50% that feel somewhat confident. So that's great. Thank you so much for completing that short couple of questions. I see that there's another question or comment in the chat. Tad says we have a number for count of beetle to FT, but not for beneficials. So I'm not sure if you have a response to that, Desiree.
Yeah,
Oh,
yeah. I imagine it's
count of beetles
per row
per
foot,
foot.
probably.
Yes.
Um, yeah, that I will admit, I I'm starting to feel like I'm saying this too often, but so much of this is so data intensive, right? Like, um, I am trying to think. Maybe I'll write down your name, Dad. and try and get back to you if I can find. I know that there has been a good bit of research done. I want to say it was in alfalfa in trying to quantify the number, basically the number of a certain pest that each different beneficial could remove. But it's so system specific. So I understand your frustration. Like, I'm over here saying we should monitor our beneficials to understand how they play into an equation. But if we don't know exactly how they play into the equation, that's a hard thing to do. Um, so I guess I'm not aware of that data personally, and I will do some thinking on if I can find it, or if it even exists. But at least what we can say is that it's awesome that you're thinking about that, to begin with, and that, just knowing that you have a beneficials present hopefully can inform your decision, even if it's not to the level of detail that we would ideally have. That's my that's my half answer. Sorry about that.
Thank you, Desiree. Okay, Kate, let's look at what's coming up for our next 3 webinars. So next week we have 2 webinars, one on cultural and physical pest control, which is on Tuesday, March 18th. The second on biological pest control, which is on March 20th. If you are looking for pesticide credits, please note that Tuesday's webinar does not have pesticide credits while Thursday's does. And then our final webinar in this series is going to be on March 25th, which is the role of chemical control and formulating an IPM plan. Next week, Samuel DeGray is going to be presenting the webinars as Desiree is going to be out on medical leave for that injury that she sustained. And so Desiree, thank you so much for being here this week. We know we really appreciate Samuel being able to step in next week, and then hopefully we'll be able to see you back here on the 25th.
I should be back then. Let's hope.
Let's hope. Okay, thank you everyone. Have a great day. This webinar and the slide handout will be posted. Desiree will move forward to getting your pesticide credits registered, and have a great weekend. We will see you next Tuesday.